Hi and welcome. I'm Irene at the Department of Twin Research. We're going to be talking about epigenetics today. This is Kirsten. Kirsten, would you mind telling us what you do within the department, please?
So my role within the department is as project manager for our big epigenetics project, which is called Epitwin.
Mm-hmm. So I'm going to ask you some questions on epigenetics, if that's okay.
Yeah, that's fine.
So I've been hearing about epigenetics as this new thing. What is it exactly?
So as geneticists, we've known about the genes and what their role is in disease susceptibility for a very long time, and we've also had an understanding that the environment has an impact on your disease susceptibility as well. And epigenetics is sort of a combining of both of these things. So what we've been seeing is that genetically diseases are heritable, such as myopia, which is about 90% heritable within families. And what we're seeing is that actually the environment is having an effect on the gene. So even though through DNA profiling the gene looks the same, there are actually chemicals that are tagging onto the gene that change how it works and functions.
So how does epigenetics work?
So as I've mentioned, the environment is actually impacting on how our DNA is working. And this can be through a whole range of different pathways and mechanisms, of which most we don't understand at the moment. But it's thought things like smoking, excessive alcohol consumption, and a great deal of stress throughout your life can actually make these chemical tags tag onto the DNA. So it's mainly thought to occur in the uterus when you're in your mum's womb, as well as in your early childhood. By the time you become an adult, it becomes a bit more stable. So it's thought to maybe an epigenetic change is thought to occur maybe over a 5-year period of time. So it wouldn't be like the second you had your first cigarette, you'd have an epigenetic change. It would take 5 years for this process to happen.
Okay. Is epigenetics a new science?
Relatively new. It was actually a phrase that was coined by Conrad Howell-Waddington back in 1942, before anyone really understood how genes worked. And he felt it was the interaction between the environment and the DNA and how that work together. But it's only recently that we've actually had the technology to be able to look into the chemical tags to work out what's happening.
Does this affect the way we understand genetics?
Well, everything is part of a big jigsaw, and we're trying to put this jigsaw together so we can find the answers to why people get certain diseases. And this certainly helps with the nature versus nurture argument, and actually pinpoints that we're not actually just a sum of our DNA, because there are people that would argue that everything is due to your genetics and there's no other impact on your life. So with these new technologies, we're now being able to show that the environment is having an effect on your DNA and changing how your DNA works, and therefore is potentially changing your susceptibility to getting diseases both genetically and through your environment.
So what is the Department of Twin Research doing to look at epigenetics?
So for anyone who's been on our website, they'll have seen the phrase Epitwin, which is actually the name of our project, which is concentrating on these methylation changes or epigenetic changes in our twins. And for this, we're actually looking at 5,000 of our twins, both our identical and our non-identical twins, and we're looking at the changes that are occurring in them, in their chemical tags. These are changes over time, so we're looking at a period of maybe 15 years to see what's happened over that time. And we're also looking at what genes have been turned off or silenced by the methylation process. So the methylation process is a naturally occurring thing anyway, and it has a good role to play in growing up, because this is what stops you from growing tall. It can be a tag that joins onto a gene that's associated with height, and that tells that gene when you're a certain age that you should stop growing taller, so that we're not all 18 feet tall. Unfortunately, it can also have a bad effect, and that can be if it joins to a gene such as a tumour suppression gene, and that actually stops that gene working and suppressing tumour growth, and therefore you would end up with a growth in a tumour.
What are the future plans for epigenetics within the department?
So as I've mentioned, this is a huge project, and we're looking at 5,000 of our twins, which is a lot more than most people have looked at in methylation projects. And with this, we have a wealth of data that we can look at. We can look at how these epigenetic changes over time are influencing things like our lung function or our blood pressure. We can also see whether there's any differences before or after you get a disease, so maybe before your onset of diabetes diabetes, and then maybe after you've actually got diabetes. We can also see what's happening between twins who are discordant for a disease such as heart disease, where one twin would have heart disease and one wouldn't have heart disease. We have the opportunity, as some of our twins in our database have twin children in our database, to look at what's happening through the generations, so what's happening between mother and the child. We also have sets of twin brothers and sisters where there's more than one set in a family, so we can see what's happening within the same generation. We can also see the differences between identical twins, non-identical twins, and single pregnancy adults to see if there's any epigenetic changes. So with our identical twins, we'll have a very similar inter-uterine environment, completely shared genetics, but a different adult environment. So we can see whether there's any influence of the adult environment more so than the childhood environment. With our non-identical twins, we can see a similar shared inter-uterine environment, slightly different genetics and a different adult environment. And then with siblings that are from a single pregnancy, we can see that there's only slightly shared DNA, no shared inter-uterine environment, and completely different adult environment. So we can look at this from all different angles. We also have the opportunity to look at twins who've been adopted and raised apart and compare those to twins who have been brought up together. And as we have more younger twins joining, we have the potential to look at the difference between naturally conceived twins and twins that have been conceived via infertility treatment, as well as looking at all the different lifestyle information that the twins have given us over the years, such as their smoking and alcohol consumption, and see whether that's having an impact on producing any epigenetic changes that we can note.
The Horizon programme was very interesting to watch, the truth about fat. So if I was a twin, how would that affect me? I mean, will I be destined to be fat or thin depending on my epigenetics?
Not necessarily. Obviously, as we've said, it shows how the environment is changing how your DNA is working. We weren't standing there and saying that in every twin pair there will be one fat twin and one not fat twin. The idea is that we're trying to put all the pieces of the jigsaw together to answer what's happening in general with everyone, so that we can look at the similarities and differences between everyone, not just our twins.
So does changing my lifestyle and my diet affect my epigenetics and whether or not I would get a certain disease?
The possibility is definitely there, and that's why we're looking at all the lifestyle information that our twins have given us over the years. And very importantly, we have a food frequency questionnaire that we're going to be able to use that's also going to help us answer these questions, and hopefully we'll complete the jigsaw as much as we can with this project.
Thank you, Kirsten, for answering all these questions on epigenetics. It really has helped me understand it a little bit more, and I hope you found it interesting, and thank you for watching.